Dear students, in the last lecture we had studied Golgi complex. I'll give you a quick revision of what we had studied in the last lecture. Thereafter we'll begin with the functions of Golgi complex. Golgi is a membrane-bound organelle. It was first discovered by Camillo Golgi, and he stained the cell of Barnouple nerve cell. That was a Barnouple nerve cell, and he could find membrane-bound organelles which are also called as gamma cytomembranes. And I told you that the marker enzyme of Golgi complex is glycosyl transferase, and it is absent in prokaryotes but present in eukaryotes, especially in those cells which are actively indulged in secretory activities. Wherever the Golgi is present, it is surrounded by a clear cytoplasm which does not have any kind of organelle, and that clear zone is called a zone of exclusion. Then come to the structure of Golgi complex. They exist in 3-4 forms. Flattened sacs, they are called as cisternae, and thereafter large transition vesicles which are having dimension of around 400 to 800 angstrom, and they are majorly found on the forming face of the Golgi, which is convex. I told you in the lecture. Then large vacuoles, they are around 600 angstrom, and majorly they are present on maturing face of the Golgi bodies. Secretory vesicles, they are present on the maturing face of the Golgi, and these secretory vesicles, they ultimately get converted into zymogen granules. Then these later on form lysosomes. You can view the structure of Golgi. The above surface is forming face, which is also called as proximal or cis face, and the face which is towards the plasma membrane is called as maturing face or distal face or trans face. Then let us come to the function of Golgi membrane. The prime function of Golgi bodies is secretion, and they secrete proteins. But it happens in a phased manner. The first stage is called as ribosomal phase. In this phase, the proteins, they are synthesized on the ribosomes which are present on the membrane of endoplasmic reticulum. Following ribosomal stage is cisternal stage. Proteins formed in the first stage are transported into the lumen of endoplasmic reticulum. This is called a cisternal stage. After this, intracellular transport of the protein which is contained inside the cisternae into transition vesicle, which fuses to form Golgi cisternae at the forming phase. Then the enzymes, the proteins, they get concentrated and are stored in secretory vesicles, and ultimately through the process of exocytosis, and these get discharged outside by fusion with the plasma membrane. This phenomena is displayed vividly in goblet cells, mucus cells, tear, and saliva. Then another function is synthesis and secretion of polysaccharides. The process is called as glycosylation. The proteins which are released from the endoplasmic reticulum are combined with the carbohydrates to produce complex carbohydrates. They are termed generally as glycoproteins, mucopolysaccharides, and glycogen and glycolipids. Both Golgi complex and endoplasmic reticulum, they contain glycosyl transferases. These sugar may be attached to polypeptide chain in endoplasmic reticulum cisternae even while the chain is attached to endoplasmic reticulum. This chain is later on transported to Golgi and additional sugars are added to the chain or sugar units already attached. This is called as glycosylation, and it necessarily comes out from this that glycosylation thus begins in endoplasmic reticulum and gets completed in Golgi bodies. The glycoproteins so formed are released into the lumen of cisternae. For example, the synthesis of thyroglobulin of thyroid gland, sugar mannose incorporated in endoplasmic reticulum, while galactose and fructose in Golgi complex in the formation of thyroglobulin. Another function is sulfation. Golgi contains an enzyme called as sulfotransferases, therefore it takes part in sulfate metabolism. Golgi also contributes information of plasma membrane because plasma membrane is renewed by the exocytosis of secretory vesicles. Golgi complex contributes plasma membrane of cells resulting in cell division. Golgi complexes involved in cell wall formation in plants and packaging and secretion. The very important part of sperm, which is acrosome, is formed by Golgi complex. Then ribosomes are also formed by Golgi complexes, as at the maturing phase we have studied the secretory vesicles vesicles which get converted into zymogen granules, and these zymogen granules later on converted into primary lysosomes. The Golgi bodies are also involved in neurosecretion and formation of retinal pigment. One of the major functions which is associated with Golgi is glycosidation of lipids and proteins to produce glycolipids, and glycolipids have polypeptide chains which are attached to glycosaccharides. Most of them have 5 saccharide units of mannose and N-acetylglucosamine. So students, this is all in Golgi complex. Let us start with lysosome. Probably today we will not be able to complete lysosome, but let us begin with lysosome. Lysosome, if you could remember, let me take you back to that slide which showcases the structure of Golgi. Look at this. The upper portion is forming phase and the lowermost portion is maturing phase or trans phase. Now at the trans phase you can see secretory vesicles which are also called as zymogen granules. These zymogen granules, they contain a battery of enzymes which are hydrolytic in nature, and these vesicle which have hydrolytic enzymes, they are called as primary lysosomes, and they get converted into lysosomes. And you know lysosome, how important it is. We are going to study it in detail. Lysosomes, they are discovered by Didav. In Greek, lyso means dissolution and soma means body. That means the lysosomes are such bodies which are involved in lysis of something, or the material which comes into their contact, they lyse it. Lysosome is also a membrane bound structure. It is meant for intracellular and extracellular digestion. It necessarily comes out from this that it must be absent in prokaryotes because it is a membrane bound organelle. It originates from Golgi complex, as you have seen in the last lecture. It buds off as primary lysosome. Then where do they occur? They are present in animals, abundant in macrophages and cells with enzymatic actions like the cells of liver, pancreas, thyroid, spleen, intestine, and kidneys. As far as structure is concerned, they are spherical, they are single unit membrane bound organelles made up of lipoproteins and has dimension of 0.4 to 0.8 microns, but contains hydrolytic enzymes which are collectively called as acid hydrolases. Now, what are labilizers and stabilizers? There are certain compounds which stabilizes the membrane of the lysosomes, thereby making the membrane steady and intact. Whereas labilizers, they tend to destabilize the membrane of the lysosome, and somehow these enzymes which are contained in the membrane, they comes out and digest the material around them. So the membrane is important to enzyme, but certain substances called as labilizers causing instability of the membrane, and they tend to release the enzymes. The examples of labilizers are vitamin A, B, E, K, progesterone, testosterone, and beta estradiol. They destabilizes the membrane of the lysosomes. But contrary to these substances, we have certain substances which have stabilizing effect on the membrane of lysosomes. For example, cholesterol, cortisol, vitamin E, chloroquine, antihistamines, and heparin. They are stabilizers, having stabilizing effect on the membranes of the lysosome. Lysosome doesn't occur in single form but in multiple forms and exhibit the phenomena of polymorphism, and it happens due to association of primary lysosome with phagocytosed material. So primary lysosome is the lysosome which is newly formed storage granules and has no association with any of the phagocytosed material or food around like that. Then phagolysosome, which is also termed as heterolysosome. When primary lysosome gets associated or merged with phagosome, it is called as phagolysosome. Another kind of lysosome is autolysosome. Autolysosomes are the lysosomes when lysosome associates itself with the organelle of its own cell, like mitochondria, rough endoplasmic or smooth endoplasmic reticulum, or any other organelle. And this mechanism, and it either kills that organelle or digests by digestion through its acid hydrolases. And this autolysosome is very important as far as growth, repair, and removal of damaged cell components are concerned. Then we have residual body, another form of lysosome, which is also called as stylolysosome, and this is the lysosome in which there is digested material and the remnants of the phagosome whose material has been digested. So today we'll end over here only. In the next lecture we'll continue with the chemical nature of the lysosomes. Till then, thank you so much.